So before I discuss the results and state the conclusions of this study, I wanted to more explicitly outline some of the connections between the methods in this work and those that we have and will continue to perform in 551 Lab. So first and foremost, our 551 Lab has revolved around this central motif of making a mutation, cloning that mutation into a vector, and then being able to purify that plasmid DNA from other DNA. This is also a vital part of this work, especially given the amount of mutant variants that had to be made to express the full saturation mutagenesis library. So in saturation mutagenesis, as was outlined, every amino acid was substituted at each of nearly 200 positions throughout the amino acid sequence. So this required a steady dose of making a mutation, amplifying that sequence with PCR, cloning that sequence into a plasmid, and then purifying that plasmid from other DNA. Just as importantly, that plasmid also then had to be introduced into cells because we want to express that viral spike protein, not just design the sequence. While we've been working with bacterial cells in lab and thus focus on transformation of that plasmid DNA, this work used human XP293F cells. So given these are human cells, the genetic information is transferred via transfection instead, but still a very similar method of exchanging genetic information to that of the transformation we have been simulating in lab. And additionally, just last week in lab, we utilized the EBI amino acid sequencing tool to score sequences shared across several different variants of human carbonic anhydrase 2 to see which amino acid positions were conserved throughout those different variants. This same technique was also used in this study with regards to the viral spike protein sequence among 7 different beta coronaviruses. And interestingly enough too, that sequencing information and those conservation scores were then used to derive a phylogenetic tree, which was very similar to that of last week's presentation as well. So this work makes very apparent that controlling the interaction between the viral spike RBD and the ACE2 receptor is vital to further therapeutic methods of controlling SARS-CoV-2. Across multiple different existing beta coronavirus strands, the designed soluble decoy receptor showed tighter binding affinity and increased binding preference as opposed to the wild-type soluble ACE2 receptor. And this is consequent to the fact that this decoy was designed after further experimentation showed certain consistent motifs throughout the RBD, such as the highly conserved hydrophobic core of the RBD, and knowing that the exposed interface surface of this RBD was more mutationally tolerant, but still maintained a certain level of conservation. Even among the surface interface mutants, the soluble receptor decoy is still much tighter than soluble wild-type receptors, and even those mutants that showed increased binding preference for the wild-type receptor bound to the decoy receptor with increased strength. So these findings are very promising for the future of non-vaccine oriented treatment of SARS-CoV-2, and up to this stage of experimentation appear to be more versatile and mutationally tolerant to other experimental methods such as monoclonal antibody use. Future directions where this work could continue to move include possibly applying this knowledge learned of soluble decoy receptors against other viruses than SARS-CoV-2. Decoy receptors have been used against some cancers and continue to be used against them, but it'd be interesting to see if this technique could apply to both future viruses as well as existing viruses like certain strands of the flu. And if it is not applicable to the flu or other existing viruses, trying to understand what makes a virus more or less susceptible to decoy receptor repression. Another angle would be to apply what was learned in this study to monoclonal antibodies. So even though monoclonal antibody cocktails seem to be more effective than singular monoclonal antibody use, there still are worries, like I stated earlier, about the efficacy of monoclonal antibodies against antigenically unique strands that could come out of wild animal infection and mutation. So could a hybrid monoclonal antibody decoy receptor construct be made in a way that blocks viral binding to ACE2 receptors while also triggering some sort of targeted immune response? Because this would seem to be the best of both worlds. Also, as research continues to move to clinical settings, it will have to be seen if this approach would require targeted administration, like for example to the lungs of a patient, or if this method could be used more diffusely and could be injected, say, in the arm, like that of a vaccine. So finally, some questions for you guys to consider after this presentation. For one, considering the findings regarding the conservation of certain segments of the viral spike protein and the increased mutational tolerance of others, why do you think we don't see the inverse? Moreover, why is it less likely that the core of the RBD is more varied as compared to the interface surfaces? Additionally, why is it important to control for avidity effects in studies like this that measure binding affinity? Why can't we just measure overall binding as opposed to specific binding that limits for these effects? How promising do you think soluble decoy receptor use is compared to vaccination? Are there any advantages to decoy receptor use over vaccination, or vice versa? And given a hypothetical future pandemic, do you think it would be more effective to put even more resources into pharmaceutical approaches or to favor non-interventional methods to stop the spread of the virus, i.e., as we've seen over the past year, social distancing, masks, or other methods?